Professor Christian Jobin is a professor of medicine at the University of Florida and a program leader for the Cancer Microbiota and Host Response at the University of Florida Health Cancer Centre. His work is dedicated to understanding host-microbe interactions in the gut and teasing out the host immune response to microbes during health and in disease. Thank you for taking the time to chat with us today, Professor Jobin.
My pleasure.
First off, can you tell us a little bit about yourself and your main area of research and how you began in the field?
Right. So beginning in the field. So I guess I started as a postdoc in 1993, end of 1993, and the disease I was studying is IBD, inflammatory bowel disease. So naturally, I was drawn into the gut and all the little dirty things there is in the gut, including these bugsy. So I got to be interested in how these microbes may be important for disease initiation. And that's a little bit how we started. And the question I was over the year that I wanted to address is how the inflammation, as you see it in chronic inflammation, may be leading to cancer. And since we knew that microbes are important to initiate inflammation, then the question became, how come or will any bacteria that induce inflammation will have the capacity to induce cancer? And during the course of these studies, since inflammation could be a different location in the gut, we were trying to tailor or study microbes that induce inflammation in a specific site and see if it will overlap with cancer. And so during the course of these investigations, we start finding interesting stuff that led me to explore and stick longer in the field of microbiome. So that was not a hit and run. That was more of a long run dedication to the microbiome.
Yeah.
But at that time, there was not microbiome. There was just host microbe interaction. Microbiome was used when the heavy sequencing and next generation sequencing came about, and then you could study community, and then we rebranded as microbiome.
As the technology progressed, you just started to rebrand.
Right, but at the beginning it was just microbes and bacteria interaction with the host.
Yeah. And do you feel that you're circulating back to that now? You're teasing back those interactions, pulling it away from the microbiome in general?
Right, no, absolutely. I mean, the person in the lab, we widen a little bit the question. It's still related to inflammation and cancer. Yeah. But now we include interaction with, diet, because that's one of the factors that influence cancer and inflammation. Therapeutics, because obviously we need to treat this disease. So are the microbes a partner or a partner in crime or a partner in therapeutics? So that's a little bit the widening of the question here, but it's still centered around inflammation, cancer.
Right. What do you think is the most exciting aspect of the microbiome research frontier in your area of research?
If you look at, flagship journal, the top 1% journal and what they are publishing in the field, I think is very interested in how therapeutics intersect with microbiome. So as a way of treating patients, do we need to incorporate microbes as the same way we're doing with genetics and precision medicine? Should we introduce microbes as part of the equation for precision?
Yes.
So I think that's what is certainly on the translational aspect. I mean, if pure academic questions, any question could be, very exciting for one investigator and maybe not for another one, but
for another one.
In terms of translational impact and the space of pharma and companies and academic, I think, therapeutics is certainly a hot field of research.
Absolutely. When do you think patients will actually be able to benefit from the current microbiome research in mainstream medicine?
Mainstream medicine. Well, I mean, mainstream medicine. It depends on the definition of mainstream medicine. I think the therapeutics aspect came from, in my view at least, came from the fecal transplant and the therapeutic impact of fecal transplant on C. difficile recurrency.
Sure.
And I think that field is more mature for translation and getting a therapeutic, whatever aspect of therapeutic that will be, probiotics or cocktail designer, it will be available within this decade, that's for sure. I mean, there's phase 2 and phase 3 clinical trial on different designer cocktail that will seems at least to prevent recurrency of C. difficile. So that will be probably the closest we could get in terms of microbiome translational impact in the clinic.
Right, within this decade.
Within this decade, for sure. If we're phase 3 right now, it's going to happen.
Yes. Okay, and you feel like the rest of the research is going to catch up in the next decade, or?
Well, all the diseases are lined up. I mean, everyone is in the space of the traditional intervention, a traditional way to intervene on disease like IBD was probiotics. But now we're going to understand that we need a little bit more understanding of what we put inside the gut is doing to a community and the disease aspect. So I think IBD is certainly the next. There's clinical trial. There's clinical trial pretty much for everything right now.
Yeah.
Cancer, autism, neural disbalance. But it took a long time because the C. difficile, the proof of principle was there with fecal transplant. We don't have it for other disease. Fecal transplant and IBD is, the meta-analysis suggests there may be an impact. The effect size is not that strong, but for C. difficile, we are talking about close to 90%. So trying to reproduce that makes a lot of sense. For other disease, we don't know. Maybe there is an application, and maybe there will be an effect, beneficial effect, but to which extent we're going to see that effect. How powerful would that be? We need to look at that, but it's coming.
It's coming. What do you see as the greatest challenge in microbiome research?
Money. Well, I mean, I think, we've been very wide in the field of microbiome. So a lot of disease were studied for association, every disease in the book, and we were not going deep. So now we start drilling and then we start hitting, the resistance, the pocket of resistance. How do we get deeper? And one roadblock in microbiome is certainly understanding gene function. So if we start doing association or we start putting new bugs and we see an effect, how do we explain these effects, beneficial or deleterious? How does it work? And a lot of time you need genomics to really understand how these microbes is doing XYZ function. And to understand genomics, you need to manipulate genes. And unfortunately, a lot of these microbes are very difficult to genetically manipulate. So this is difficult for many laboratories.
Money.
So there's no toolbox. To you have XYZ microbes, and here's how you're gonna manipulate the genome to confirm that what you found is responsible for the effect. And not clear. We don't have that. I'm not talking E. coli and those traditionally very easy bacteria. I'm talking about difficult bacteria, first to culture and second to genetically manipulate. So we need to solve that, and different lab has built up tools, but they are not movable to every species on the book. So it's really like a cowboy. People are doing it on their own and hope that we will have enough material and I guess not material, but approaches to start answering these very important questions.
Is there an alternative to the cowboy approach, do you think?
Well, I mean, resources, and people get funded through consortium. So if this question is, hey, we're going to build a toolkit to manipulate, the top 20 or whatever you define, which is the most difficult bugs that we see are important to study, but we don't have ways. So, you will have to put money, and people will start working together, try to solve this. Right now is just single lab doing things on their favorite bugs, and they drill it and then publish it. Then another lab doing it. There's no concerted effort that I know of. There's consortium right now to study function of bugs and different diseases, but I don't know. They are not dedicated to a resource on how to manipulate genes.
Fascinating. If you, and this leads into the next question, had unlimited resources, so it fits right nicely into what you've just mentioned, what scientific question related to the microbiome would you most likely explore?
Limited resources, money, and people.
Yeah. What questions could you answer?
Yeah, no, absolutely. I mean, there's so many questions, but I mean, at this point, we have a very tunnel vision. We start with community. We study microbiome community, like which one is associated with disease, for example, and we lock in on bacteria. Or another group is viruses. Another group is fungi, and we silo that. But we are ignoring that this is a community that lives in a space, and they share, they cross-talk. So this inter-kingdom conversation they are having between them and us, we are ignoring that. We don't have the capacity to study it or the bandwidth and the resources. So you just take a kingdom, so let's say the bacteria, and then you go down and you study their function, and you narrow it on one or two species and so on. But the community view and inter-kingdom relationship between virus and bacteria, fungi, as a whole, and how they work to impact us, because they do. It's not only one type that will impact. So we don't have this system biology view of the microbiome. And, if someone will have to have that multiple resources, it could try to tackle these questions, but those are, requires a lot of resources.
Yeah.
So,
hopefully one day. What's the best advice that you could give to anyone on how to best care for their own microbiome?
Well, look, that's interesting, because I'm eating these nice little for breakfast, actually, almost veggies, but that's interesting. That, and I'm crunching in the microphone. But the idea, let me think about it. So
starts with your diet.
No, I mean, that's it, exactly. So we know from decades, we don't need the microbiome. We knew and we know that diet, exercise, good sleep, reduce stress are all important element of a healthy, holistic life. And then along the way, right, the last 10 years or so, oh, look at that, stress is impacting microbiome. Oh, look, exercise and diet, and so you don't need to be taking probiotics XYZ to take care of your microbiome. And you may, if you take care of yourself, it will reflect on your microbiome to work with you to maintain health. So it's a very holistic ensemble of body and microbes that live together. And so the short answer will be treat your body the way that we know we should treat it. Good diet, exercise, sleep, reduce stress, and and if you look at the microbiome, it will smile at you.
Yeah.
Right.
So you want to essentially nourish your microbes with doing all that. Why do you think people are reluctant to sort of adopt these very simple, basic, lifestyle choices, I guess, to sort of nourish their microbiome? Like, the answer's there. Do we need the science to catch up?
No, well, we're lazy people. I mean, in general, we want a pill. We want a little thing that we take every day, a supplement that maintain your health, and you don't make the effort toward because it's difficult. I mean, it's a simple concept, but compliance on a diet change is is terrible.
Yeah.
So yes, you could accomplish it, for New Year's resolution, and then how many weeks you're gonna last, and exercise is the same. So it's really a lifestyle and a paradigm shifting in how you lead your life. And I'm not the flagship of how you should, live your life.
Well,
but
you're
I'm
demonstrating.
saying,
You're demonstrating, how to do it right now, which is fabulous. I saw
oh
some
no, that's
carrots
for
in
sure.
there and
That's for sure.
vegetables.
I have my weak moments though.
We all do. I think that's the microbes in your soul rather than in your body.
Right. That's right.
The pleasurable moments. I'll go on to the next question, and I would like to know, where do you see the microbiome research in general being in 5 years from now? And you spoke about the technology, catching up. Where do you think it's heading in 5 years' time?
Right. No. Yeah, for sure. In terms of microbiome, we need to be more granular, and we are doing it. Some fields are more advanced because they started asking the question earlier than other discipline. But anything that goes with metabolites, biotransformation of dietary compound, of drugs, or interaction with the external cues that we receive, all these capacity of microbes to process these information and output something. So, if you take XYZ drugs, how is it that, or how the microbes may be transforming this drug for a good output or a negative output. Any good diet that should produce XYZ compound, but doesn't happen because you don't have the right microbes to do it. So all these interaction at a detail, more granular level. So metabolism, microbiome derived metabolism, I think is very important. And there's a couple of labs that does that, but it's not wide. It's just a pocket of labs. But I think that will come bigger. And if you look at that, there's many labs now around the world. Microbiome genomic, it becomes like a Western blot of the Southern blot, Northern blot of the 80s, because technology is there, companies are there, you can send your samples and do it. We don't have that for heavy metabolism of microbes and genetic editing of microbes. So it's still cowboy type of research. But as technology companies sees, hey, this is a pocket, we could fill that gap. They will feed on investigators that are showing tools and how to do it, and then these companies will come say, we could do it for you, and then so there's more people, and so the field can become more vivacious and less of a unique technology driven lab or a couple of labs driving the field. So it will become more, let's say, democratic.
Tell me what your personal highlight of your own microbiome research career has been so far.
Well, is to look at my microbiome and say, hey, you're not that bad after all.
Have you sequenced?
We sequence everyone in my lab to look at, for whatever metric we're using, diversity and all that thing. That was interesting. No, but I'm kidding. So, but it was something to look at it. No, I don't know.
What was your reference for it, though?
Well, we had, all the people in the lab. So we were like 20, and then spouses, then kids, then dogs, and,
wow,
everything that is so. And then we did the PCA, and then we look at diversity and richness. And so we could heat map richness, the more diverse and rich microbiome, and who is not on the PCOA plot. So we could see people that are less diverse and some that are very diverse. No one with a underlying disease per se. Everyone is healthy. So that was a very interesting conversation to have about what are you eating, why are you there, and how could you be that diverse? I want that diet, So that was an interesting conversation.
Wow.
But it was just for the sake of and we print T-shirt actually of this microbiome. Everyone has his own microbiome printed, so it was interesting.
What a fun little thing to do, but very revealing.
No, that was fun, and it bring awareness because you study this, and but no one has really, you know, scooped their stools for microbiome analysis, which is a very personal thing to do.
That's
So
right.
that was interesting. It
Is it
brought
a pre
awareness of,
is it a
yeah.
prerequisite to join your lab to have their
No, I did not made it. Actually, some people didn't want to do it, and I respected that. But that's going to be an interesting thing. A waiver. I'll sign. You know, you have to sign that you will donate microbiome for the cause.
Yeah.
But I think long time ago, I think that, you know, when I studying the microbiome, not microbiome, but bacterial host interaction, and then we started in 2007 or so, 2008 to sequence, and I didn't know if I was to stay there, the genomic, because I thought it was very, you know, counting stars, and you know, I thought it was too superficial. And if you're coming from the field of signal transduction with all these phosphorylation, interaction with molecules, dissection, precision, I thought that it will be boring just to sequence and get bugs. But then as we start getting more involved in the sequencing and then isolating or identifying natural product from bacteria that cause DNA damage. Now I saw the straight pipeline, the flow chart from genomic, descriptive to candidate to gene function to outcome on the host and cancer. And looking back, that was discovered that in 2010, 11, published in 2012. And the field has advanced, and now I see labs identifying a signature of this compound in human cancer. So you see that. So it's very rewarding that the field is moving forward.
So you made the right decision.
Well, yeah, no, absolutely, but also that the field, it has meaning. It's not just to play, have fun, but it could be impactful for patients. So it's rewarding that you have fun doing research. At the same time, it may have a very important impact on patients. So it's not just you know, raising money and hiring people to intellectually answering rewarding question, but it will impact
lives,
clinic, lives.
If you didn't choose a career in medical research or scientific research, what career would you have chosen?
Oh my gosh! I mean, I came from this space of being an artist. I chose science for the artistry of it. So the freedom of creation. Yeah. And you know, I would have probably been doing something else related to art, I guess, right? And because I just don't want the confinement of you know, of a job that restrict you in your mind. So that's what science was so appealing, and still is.
Well, Professor Jobin, I totally understand why we have nominated you to be part of the interview web series for the MRC. You are truly a maestro of the microbiome. Fascinating that we've had.
Too much.
No, I sincerely mean that. And I think we've had such a fascinating chat, and I could probably, you know, keep going with a lot more questions. And I think
but it's late.
Yes, I learned a lot from you, but it is close to midnight. That's correct. But what I'd like to do is just to wrap up our interview and our time with you today. And I look forward to having a chat with you at some other time. And I truly believe, yes, you are a maestro of the microbiome. So thank you for your time.
Thank you, Fatima. It was great talking to you. Thank you so much.
Thank you.